participants. Our next and final speaker is Michael Farkouh, Professor of and Vice Chair of Research, Department of Medicine at the University of Toronto.
Thank you, Dr. Altman. Again, it's a pleasure, I echo what others said, to be here today. And I'm representing a number of constituencies, most importantly the Mayo Clinic, which are my partners in this clinical trial, and they actually provided the first $3 million of support for this clinical trial of clopidogrel pharmacogenetics. It came from their new Center for Individualized Medicine. They've done a fantastic job on the cancer end, and they were very intrigued and very involved in engaging the cardiovascular community. And I serve as the advisor and mentor to Naveen Pereira, who's actually the administrative PI. He could not be here today, so I'm representing Naveen and our steering committee today on this trial. This is a collaboration between the Mayo Clinic and the University of Toronto. I will say that it has developed into a number of very unique partnerships. Halfway through the trial, we did get NIH funded, and I'll talk about that. This is a 5,200 patient trial. At about the 2,700 mark, we learned of the participation of the NIH, which was very, very important to us. We also engaged a lot of our academic partners, both in Canada, Korea, and the United States, total of 29 centers. We engaged industry partners, not the ones we thought we would. We engaged a platform, new company that does point-of-care genetic testing called Spartan Biosciences in Canada that measures a point-of-care for the CYP2C19 loss of function allele. And we did try to engage pharma in this because there are two other drugs that are used in this field, and ones in which we pursued, of course, industry sponsorship, but on multiple occasions it was refused. So that's something of a lesson that we'll go through in a moment. I have nothing to disclose. The objectives here are really to talk about clopidogrel metabolism, to talk about the genetic variations in CYP2C19, which metabolizes clopidogrel into its active metabolite, and then the design of the Taylor PCI trial and some of the obstacles we faced along the road here. So in the Journal of American College of Cardiology, which is our leading journal of cardiovascular medicine, there was a comment in editorial, one of the most important unresolved issues in interventional cardiology is this issue. But if you ask most cardiologists, they're not thinking about this because we've moved on to newer agents and we've moved on to newer and bigger problems. So this remains a problem, and I'm going to try to convince you why this still is important to the cardiovascular community. Clopidogrel is used in patients with vascular, atherosclerotic vascular disease. We know that coronary artery disease affects almost 10 million folks in the United States. For clopidogrel alone, at its height in 2010, there were 2.5 to 3 million prescriptions written per month. It was a $6 billion compound before it went generic a few years ago here in the United States. And we know that it has, as I said, multiple indications for atherosclerotic disease, including myocardial infarction and acute coronary syndrome with stenting, thrombotic stroke, peripheral arterial disease. So as I said earlier, clopidogrel is metabolized to its active metabolite through the cytochrome P450 system. The active metabolite binds irreversibly to the P2Y12 receptor on the platelet. And so it has been shown now in dual antiplatelet therapy, aspirin, clopidogrel, to be very effective in the treatment of acute coronary syndromes in patients having coronary stenting performed. As I said, there are new agents which I'll talk about in a moment. The genetic variation in CYP2C19 is very interesting. It follows, there are two loss of function alleles, star 2 and star 3, and there's a gain of function allele, star 17. For the study at hand, we're really focusing on the loss of function allele, which is star 2, star 3, which we can measure in this point-of-care testing. And you can see there's ethnic variations in the polymorphisms. So in African patients, 15% have star 2, all the way to 30% or so in East Asians. And then star 3, also less prevalent, but also associated and varied across ethnic groups. So one of the challenges in the study was to incorporate folks from different ethnic groups so that we could enrich the population in terms of those that had the polymorphism. Now, one of the big questions going into this was, does the genetic variation in CYP219 affect clopidogrel outcomes? And there was a very effective meta-analysis by Jess Mega, which took into account all of the major studies of clopidogrel in which the genetic polymorphisms were measured. This was involving almost 10,000 patients across a number of clinical trials. Patients were broken down into those that were at genetic risk, had loss of function allele versus those that did not. And you can see in its totality that there's about a 57% increase in risk of CV death, myocardial infarction, or stroke in patients who have loss of function allele. So certainly there's an epidemiologic basis to address this question. This led, and again, here in this study, about half the patients had acute coronary syndromes. The others were stable coronary patients, but 91% had a PCI performed, many of them with a coronary stent. And it was shown that 1 in 3 patients developed an ischemic event if they had loss of function allele. So there was a lot of hoopla about this and a lot of interest. Is this really an important thing that we should be measuring? And I have to say in the background of this, because there's a huge body of work that I won't cite, that in terms of its effect on platelet function, there hasn't been so much, the studies have been negative for the most part. So this now is really focusing on the genetic variations and not on platelet function. This led in March of 2010 to a black box warning by the FDA stating that the effectiveness of Plavix or clopidogrel depends on activation to an active metabolite, and that poor metabolizers exhibit higher cardiovascular events based on the evidence we showed. And tests are now available to test the genotype, and perhaps we should be recommending this. Is this a black box warning? And this was something that the cardiovascular community grappled with, because the issue is, are we going to start, in patients who are on clopidogrel, are we going to start measuring the genetic polymorphisms? And there was a consensus document from published in Circulation from the American College of Cardiology and the AHA, led by David Holmes, in which they stated the following: the evidence base is insufficient to recommend either routine genetic or platelet function testing at the present time. There is no information that routine testing improves outcomes in large subgroups of patients. And for the most part, this is where the cardiovascular community has continued to live in terms of this question. But it has hit the press. It's hit the Wall Street Journal, and it's hit the other major journals in the field of business and so on, is saying that there's a dilemma here, that maybe this is challenging. There are studies that both support what the FDA did with the black box warning and then refute it. So it's become something that's been topical. Again, our patients ask us about this, and the community has continued in terms of guidelines, which have continued from 2011, to give it a class 3 recommendation of no benefit in terms of measuring the polymorphisms. It states the routine clinical use of genetic testing to screen patients treated with clopidogrel who are undergoing PCI is not recommended, so a class 3 recommendation, and that's where we stand till this day. Complicating this in terms of the acute coronary syndromes, we've had 2 major compounds have been developed and shown in large clinical trials to be superior to clopidogrel in terms of the major adverse cardiovascular events. One of the drugs is prasugrel, the other ticagrelor. I would just say that for the most part in the United States and in North America, ticagrelor has taken a larger share of the market, so we tend to focus on this agent. It's a twice-a-day drug, but it has also been shown in a large clinical trial called Plato to actually reduce cardiovascular death. And so it has become sort of the standard bearer. In the Canadian guidelines, actually, ticagrelor is favored as the drug of choice. But in the American guidelines, they're all class 1 recommendations for clopidogrel, prasugrel, or ticagrelor. So as the newer agents have come on board, although they're more costly, we have noticed actually in some large registries that people are still using clopidogrel for those having coronary stenting for acute coronary syndrome. It's about 70% of the market to this day is still using clopidogrel, despite the evidence on the 2 other agents. So the question then comes, with the black box warning, it hasn't gone away. It's still there. There's a gap in clinical evidence. And does altering therapy based on the CYP2C19 status affect clinical outcomes? And that's how we led to the Taylor PCI trial. actually, my colleagues at Mayo called me one day and said, you know, this is something we grapple with, and we use a lot of clopidogrel. And are we doing the right thing, and should we do a clinical trial? And so they went to their research committee and garnered $3 million of support to start the trial with no guarantee that we'd ever be funded by industry or by the NIH. So I think the Mayo Clinic deserves a great credit here for embarking on a study in which they believed there was a great clinical question, and they put their shekels on the table. So that's what led to Taylor PCI. Taylor PCI is a large pragmatic study for the most part. We screen patients who are having stenting. In 2 of the jurisdictions, in Canada and in Korea, we only screen patients who have acute coronary syndrome. And this has a lot to do with the coverage of the drug in those 2 countries. In the U.S., mostly in the Mayo system, which has been involved in about 20 centers, we have opened it up to not only to ACS patients, but also to high-risk stable coronary patients who are over 65 or who have diabetes or renal insufficiency. So we screen the patients at the time they present for coronary angiography for possible PCI. And then patients are randomized to 2 arms, a conventional arm, which is usual care, which in this case is clopidogrel 75 milligrams daily after a loading dose, and we follow the patients for 12 months on the drug, and patients are then genotyped at the end of the study. And then in the prospective arm, there's genotyping that's done, and again, we can identify the CYP2C19 star 2, star 3 with the point-of-care testing. And if patients have those loss of function alleles, they're given ticagrelor 90 milligrams BID for a year versus the wild type, the folks without the loss of function who are given routinely used clopidogrel. What's unique in this analysis is the primary statistical analysis isn't the comparison of the 2 randomized arms, but rather the comparison here. The comparison here of the folks who are on the conventional arm who got clopidogrel, who have the star 2, star 3, versus those that have star 2, star 3, and ticagrelor. This is the primary statistical comparison, which differs from all the other trials in the field. And there have been other trials that have been stopped early, and there's another trial that I'll talk about. But for the most part, that's the primary statistical analysis. So we now have a randomized clinical trial. And again, the endpoints are death, myocardial infarction, stroke, urgent revascularization. So they're fairly robust endpoints in the trial. It's a randomized trial of 1-year event rates. We anticipated event rates of about 10% overall, which is from larger other clinical trials, with 8% in the intervention group and 12% in the control group. We grappled with the effect size here. One of the challenges we had with the industrial sponsor with the company that makes ticagrelor, which is Astra, is that they felt like, well, we have shown superiority, so therefore, if we're saying that all the effects are in the genetic subgroup who have the polymorphisms, we're not going to have the market. It's a marketing issue, and that's not actually the case. What we're not claiming that ticagrelor is not superior to clopidogrel. What we're saying in this trial is that the effect size in those that have the polymorphisms will be much more. So I think that's something that was hard to get everyone's head around. So we're looking at an effect size here, not of 20 to 25%, but a hazard ratio of 0.65 or a 35% relative reduction in events with power of 80% and an alpha of 0.05. Now, the other issue that's important in this trial, of course, is it's all dependent on what proportion of the patients have the loss of function allele. And it's overall in North America about 30%. In Korea, it's as high as 50%. So that we know that in the Asian population, it can be as high as 50%. So we actually, and I'll show you some data, we're at about the 35% prevalence of having the polymorphisms in the trial. But we needed to enrich somewhat, and that's how we went to Korea with our Mayo alumni that are there, and they've done a fantastic job. There is another trial called Popular. Again, it's a direct comparison. In this trial, the comparators ticagrelor or prasugrel versus routine genetic testing for clopidogrel. And it's a trial of a STEMI population, a smaller sample size, a higher risk population. But that trial is in play as well. But these are the 2 trials that are now in the field at present. There was an NIH trial called PAPADAD-2 that was stopped for futility, and there was no enrollment in the trial. So that was one of the challenges as well, because we went to the NIH with our trial, and they said, well, why didn't you help us in the trial that had failed? And the reason there is because they had used prasugrel as the comparator, and we believe ticagrelor should be the comparator. There were some other issues as well about the administration of the trial. So again, we faced challenges to go back to NIH to ask them to fund a trial that they had turned down, or that they had stopped for futility of enrollment. So that's another thing we can talk about at the end. Some of the important milestones. At the time that we secured NIH funding, we had 2,700 of the 5,200 patients recruited. We're now at 3,700. And again, we were using this novel point of care platform. Again, all the patients in the trial in that 12 months will be genotyped. The blood will be analyzed from baseline at 12 months using routine GWAS measurements. But the point of care testing, of course, done only in the intervention group. And the lack of pharma support, I think the degree of the support, it's complete lack of support, so I want to say that. So even providing the drug to the centers, which is what we went for, which was just to provide the drug in the 30% of people in the group that was randomized to the intervention arm, and even there we did not have that. So we had to use a lot of our resources just to pay for the ticagrelor, which is in the order of about $800 to $900 a year for the course of the trial. The pilot study, we looked at the data at the time of NIH support, 2,700 patients in. You'll see here that of the patients who were in the prospective genotyped arm, 1,375. You can see here about 35% had reduced function allele, about 800 patients with wild type. So we really needed to enrich, and that was one of the reasons to go to Korea and into certain areas of Canada as well. And if you look at the baseline characteristics of the first 2,700 patients, you see they follow what we traditionally see in these kinds of trials, 62 years of age, 76% were male. We did have 66% Caucasian, 29% Asian, African American only 2%, which is too low. And you'll see here that 81% of the patients had 81% of the patients had an acute coronary syndrome. This is obviously highly representative of the Korean and Canadian contribution, but even in the Mayo system that's involved, it's about 75%. And you can see we had 26% with diabetes, with other comorbidities. 40% of the patients had multivessel coronary disease as well, which is important as how we manage the patients in the long term. So again, we secured NIH funding about halfway through in May of 2016. It's a U01 grant now. We're just finishing our first year. It's been tremendous to work with the NIH, and they've helped us on a number of fronts. One of the most important things they helped on was ascertainment of events. Because we did not have to report to FDA, or serious adverse event reporting wasn't mandated in this trial, we found that we were starting to miss events that were hospitalizations for urgent revascularization. The investigators labeled these as hospitalizations, but the NIH encouraged us, and I'm glad they did, to go back and look at all the hospitalizations and adjudicate them, because we found a number of events that otherwise wouldn't have been counted, which is a unique challenge which we had not faced before in clinical trials. So as we went large and pragmatic, we had challenges. We have an independent events committee and, of course, an independent DSMB. The NIH also adopted our DSMB, which is chaired by Deepak Bhatt. So we thought ahead of time, if we ever got NIH funded, we had to assemble the right DSMB, and to their credit, they acknowledged the DSMB that had been in place and added a few other members to the group. So overall, Taylor PCI, we believe, is dealing with a high impact, highly prevalent condition. So it's a large pragmatic study. We hope to address the unresolved practice, or if there is still the question in people's minds about the FDA black box warning. We've created a biobank for studies of GWAS, so we can do multiple other studies and analyses. And we've created a fairly robust clinical trials network now that we can work on with these large pragmatic studies. The average reimbursement in the trial is $680. So it really is done cost effectively. And we've continued when we secured NIH funding, we went up to $1,000 per patient. So it really is, done on the backs of a lot of folks and our investigators, and to that credit, we'd like to acknowledge them. So I'll just close by saying that we believe the question's alive. We think we need to address it quickly. We have another year of enrollment, so the trial will likely report in 2019. And I look forward to anyone sending any questions my way if you have any other concerns about the trial. Thank you very much.
We have time for clarifying questions.
Vicki Pratt, representing the Association for Molecular Pathology. So just for clarification, the Spartan device is not a point-of-care device. It's not classified as that, though they do market it that way. The other thing is that...
Well, it's done at what we mean. I mean, we maybe semantics here, but it's
Maybe.
done. What happens is that we
I'm very
understand
familiar
the polymorphism at the time that we're enrolling the patient into the trial.
The other thing is in the Spartan device, the STAR 2 is a Caucasian variant, the STAR 3 is a Asian variant.
Yeah.
So it's not surprising not see positivity in your African American population,
Yeah.
and that probably only represents the admixture in the African American population. So do you have any plans after the trial is over to actually look at African Americans and the variants in there, because you can actually go on
Absolutely, we intend to
GKB and
do that.
see that.
Absolutely, yeah. We've done the GWAS, and all the patients will be chronicled.
Michael, great, really interesting trial. I'll leave alone the question of marketing claims about devices versus what they actually should be for, but no comment on that one. But I'm interested in your study design where I just want to, this is just a point of clarification for later discussion. So you're randomizing testing or not, and then you're comparing within the randomized arms two groups, which technically then are not, it's not a direct randomized comparison.
Right.
You could say it's sort of inferred that they should be at similar risk. Is that, have I got that correct?
Yes. So it's taking the STAR 2, STAR 3 cohort, the loss of function, and comparing those that were in the conventional arm to the intervention. You know, Rob, we struggled with this a lot, and it went through a number of external advisors, FDA, the NIH advisors, and it was approved. I have to say that it was a unique and a challenge to get to that point. And the question is, does that...
I suspect
could
there'll
you
be
put that
some
study
unique
design up
discussions when the
real
results
quick?
come.
Yeah.
I think it's worth staring at for a second.
Let's go back. I mean, maybe we can
easy
go
to
back
do.
to
I
the
don't know if
slides.
Mel- maybe that's
Just
a-
go back to the slide with the schema, Mike.
That's time travel. We can't
Yeah.
do that here.
Back here. It's a few more back.
It's the one with the tree at the
More, yeah.
bottom. Sorry, maybe I'm the only one.
It's here.
Good.
So the primary statistical comparison, as Rob pointed out, is CYP2C19 in the conventional arm, this group outcomes versus this group's outcomes, who got ticagrelor.
So there's subgroups within a randomized comparison,
Yeah.
but the subgroups are not derived through direct randomization into two
That's right.
groups. And
Yeah.
That's an interesting.
And you're doing that because the purple is what all of our cardiology friends, or many of them, want to do anyway.
Sorry, go back one. Yeah, back one, Mike.
Purple is the de facto standard of care. That's why
Yes.
you have to have that arm.
Yeah.
Because otherwise, if you do blue only, then you're doing something irrelevant. To
Now,
the
we
current-
do, as a secondary analysis, have the comparison of the randomized arms, which would be the traditional.
But Russ,
approach.
I mean, the other point is, if the question is, should the test be done, it's really purple versus blue altogether.
Yes, and that's, yes. And that,
That is a secondary.
yes, Rob,
Yeah.
that's the major secondary outcome measure, yes, of course.
Okay, I think that ends the clarifications, and we will have a break in an hour.